Hi everyone online and welcome to this virtual presentation. I'd like to present some 7 Tesla fMRI data on volt responses in the human spinal cord at the single subject level. I am part of the research group Pain Perception at the Max Planck Institute in Leipzig, and first I would like to tell you why we are interested in the spinal cord. Our research focuses on pain perception. And pain is an important warning signal, but in its chronic form, it is also a source of immense suffering. Pain signals arise from the nociceptors and are transmitted via the spinal cord to the brain. In more detail, we know that signals are initially processed in the dorsal horn of the ipsilateral side within the spinal cord. Afterwards, the signal crosses sites and is sent via thalamic and brainstem relays to the cortex. As a research group, we are interested in the fact that perception of pain does not always exhibit a one-to-one relationship with tissue damage. Instead, pain is strongly modulated by many different factors. One way how these factors can influence pain perception is via descending pathways that influence earlier stages of the processing hierarchy. This means the very first modulation of nociceptive signals can already take place in the spinal cord. But despite its importance and potential clinical relevance, our knowledge of the human spinal cord in light of pain perception is still limited. One approach to investigate spinal cord function is to use functional MRI, but this comes with several challenges, some of which I'd like to point out here. First, compared to the brain, the spinal cord has a very small cross-section, as you can see here. Then within that cross-section, we would like to distinguish different gray matter regions, such as the dorsal horns implementing sensory function and the ventral horns implementing motor function, and of course reliably separate gray and white matter. So we need to be concerned here about partial volume effects. In order to mitigate that, people generally aim to use a high in-plane resolution, such as 1 × 1 millimeter for fMRI studies. But that obviously leads to a low signal-to-noise ratio, which is a further prominent problem. So in order to address these issues, people have already turned to higher field strengths, such as 7 Tesla, and have, for example, delineated fine anatomical details. They have characterized resting state connectivity with fMRI and have performed T1 and T2 star mapping. But obviously moving to higher field strength comes with its own problems. And I'd just like to point out a few here in the case of 7T spinal cord fMRI. First, there is no agreed upon coil design, and only a few custom-built coils exist. Second, field inhomogeneities present a significant problem, both at large and small spatial scales, such as the rapidly occurring differences of tissues with different susceptibility. And third, and this is an example I want to highlight, the impact of physiological noise is increased. So just to give you an idea, here's an extreme example of respiratory noise. And respiration causes dynamic B0 shifts and leads to apparent motion in the whole image, as you can see here in this sagittal view of an EPI acquisition. So how did we approach 7T spinal cord fMRI then? So first of all, we are using a custom-built 24-channel spinal cord coil that wraps around the sides of the neck. To correct the mentioned physiological artifacts, we additionally record respiration and pulse. To address large-scale B0 inhomogeneities, we use susceptibility-matched pads around the neck for passive shimming. In order to induce pain in our volunteers, we then apply heat stimuli via an MR-compatible thermode on the arm and use a block design to elicit robust volt responses. We use 4 runs, each containing 8 of these 30-second blocks of pain stimuli, and each block has an elevated temperature with additional peaks to prevent habituation. And the temperatures are individually calibrated so that each participant rates the stimuli as clearly painful but not unbearable. To assess autonomic nervous system responses to the pain stimulation, we concurrently record skin conductance at the hand. And our first recordings indeed showed skin conductance responses within that 30-second window for several subjects, with the strongest responses at the onset of each stimulus window. Now for the MR images. We acquired structural T1 weighted images with a resolution of 0.75 millimeter isotropic and can appropriately segment the spinal cord in here as needed for registration purposes. We use an even higher resolution of about 0.4 millimeter in plane in structural T2 star weighted images to be able to segment the gray matter of the spinal cord, which you can see as this butterfly shape here. But the real challenge are obviously the functional images. So these are way more prone to signal dropout and distortions, which we aim to mitigate with the scanning parameters listed here. Our slice stack is centered at the spinal level C6, where we would expect our activation to occur. And on this exemplary transversal EPI slice, you can see our target region, the left dorsal part of the cord. To come to the data analysis now, so we are employing the following pre-processing steps. We use a slice-wise motion correction algorithm that has been developed for the spinal cord. We include these motion correction parameters into our model and also automatically censor remaining outlier volumes. We also apply a multi-step nonlinear warping to template space and use standard statistical analysis pipelines using FSL feed. And we take care of the different sources of noise using a slice-wise physiological noise modeling as developed for spinal cord fMRI. Here you now see an example of one participant's EPI data. But we have to be honest and admit that data quality differs a lot across participants. So here you see data from another participant. And based on the anatomical scans, we know where the spinal cord should be and marked it here in green. And you can clearly see distortions or signals dropouts. For example, in this exemplary slice, you see also here below. And initially for the abstract, we acquired data from 4 participants. 2 of whom had adequate data quality and whose data we could thus analyze, and 2 of whom had clearly insufficient data quality and were thus discarded. But these distortions and signal dropouts you see here are likely due to anatomical reasons and would be reproducible across sessions. Therefore, in order to be more efficient with the participant selection, we now implemented an MRI-based screening procedure in which we invite participant once to determine their data quality and then invite those that have adequate EPI data quality and want to participate again for a second session, in which we then conduct our pain experiment. And with that, we have now acquired 70 spinal MRI data from 40 participants. And based on these screening scans, we now determined those participants who would be suited. And at the current stage, our analysis is now based on 6 subjects. And so now we could obtain 6 data sets that are of adequate data quality. And the green line again shows where the cord should be according to the anatomical images. And you can see that this matches quite well with the EPI images. So there is only minimal distortion or signal loss in the EPI data. Now to the both responses on a single subject level. As mentioned, we expect to see activation in the left dorsal part of the spinal cord. The mask we use here is an automatically created probabilistic mask from a template that was brought to subject space and depicts the dorsal left quadrant of the cord. And indeed, in 3 out of our 6 participants, we observe activation in the expected location, suggesting that task-based fMRI in spinal cord is indeed feasible at 7 Tesla. In the other 3 participants, we were not able to see such responses, although their data quality, including temporal signal-to-noise ratio, is comparable to that of the responsive participants. So interestingly, we noticed that for these participants, the individually calibrated temperatures were somewhat lower than in the responsive participants. So that might explain these differences. In summary, we have seen that fMRI of the human spinal cord at 7T faces quite a few technical challenges. But despite this, we were able to observe single subject bolt responses to painful heat stimulation in the expected region. Although it can be seen that the consistency of the responses across participants is currently unclear. And as one of the next steps, we are evaluating different procedures to address distortions in EPI data in order to allow for more fine-grained response delineation in the gray matter of the dorsal horn. And although this would require some more work, our ultimate goal would be to obtain data with such a quality that will allow us to investigate the interplay of bottom-up and top-down factors in pain perception in different layers of the dorsal horn. Thanks a lot for listening to this short presentation. And if you have any questions, just send me an email.